just yes, moderator or presenter. So now you can share your presentation.
All right.
Do you have a PDF file, Tom, or do you have PowerPoint?
I have both. I was hoping to maybe start with the PowerPoint, and then if we have trouble, switch to the PDF, because it will allow for more of a sequence for some of the animations.
Okay, let's see.
So please try to share the document so we have time to see if it works.
Slowly.
Yes, I know.
Maybe I'll move closer to the router while it's up to try to speed it up. If it will make it quicker.
Las personas, so there is it. No.
It says my presentation is at 24% uploaded so far.
Esta creo que es solo la presentación que se pone en la sala para que la gente participe.
Pero eso te iba a preguntar, ahorita la gente que está llegando a la sala no nos está yendo, no nos está viendo ni nada.
No, sí nos oye.
Ya.
Ajá.
So you're in the second day of the seminars, correct?
Yes, correct.
And everything is going well so far?
Yes, well, we expected more people, and I guess that there's been so many webinars about everything that maybe people is like choosing between a lot, and also trying to take a rest from computers, I guess. So, well, the good thing is that these are the same day that we are going to have our Congress in Guayaquil. So this is kind of a, yes, this is kind of a reminder that we are still working.
Yeah, I think the webinars they're a blessing and a curse. You know, it's.
Yes, I know.
It's that way, but I mean, it is very nice that we can have these discussions over large distances, and with some of the that we've been doing with various groups within the United Nations, I've never seen so much happen so quickly.
I know.
For
right?
global action. That's exciting.
Yes, it is.
But it can be fatiguing, right? So.
Yes.
All right, seems it's almost there.
Great.
If I may suggest, maybe you can also try to upload the PDF file and at this time, so that in case the
Yeah,
PowerPoint presentation doesn't work, you can easily move to the other one.
sounds good. So it seems it's available now, the PowerPoint. I don't know, do I need to say share?
Yes. René, para compartir solo tienen que poner compartir la pantalla, ¿cómo es?
Pero que compartir la pantalla desde la computadora del presentador.
O sea, parece que ya se le cargó a Tomás la presentación.
Ya, déjame
Entonces,
chequear a ver si está ahí. Sí, tiene que solo darle clic encima de la presentación y poner compartir. Ahora. Yo
ya.
eso lo estoy haciendo.
Ah, perfecto. Perfecto. Perfecto.
Tom, would you try to move to the next slide?
Sure.
Just to see if it works.
I'm just trying to see. So it seems like it basically converts them into a PDF. So it's fine.
Really?
It'll work. It just means that there's a lot of information all at once, instead of having it sequentially. But I think we'll be okay. We'll be okay. Oh, and then it blocked off all of Latin America in this one.
Really?
That's
why they said it's better the PDF.
I think it's probably converting to PDF.
Yeah.
But it seems overall, it seems like it's equivalent to the PDF. I think.
Yeah.
Are you hearing my audio okay?
Yes.
And,
Yes, very
yeah,
well.
Well.
Well, some of my students are here also. That's great. Thomas, do you understand Spanish? Do you speak a little bit at least?
Yeah, I understand quite a bit in Spanish.
Yeah, nice.
Not very elegant in speaking Spanish.
Great. So I'm going to present you in Spanish.
Yeah, great.
Feel free to speak in English. Don't worry. People here speaks English very well, especially people in science, you know. So
I apologize that my Spanish isn't better.
don't worry.
You don't have to apologize. Instead, thank you very much once again for your
Yes, thank you very much for accepting.
Yeah.
So let's see. Let's wait. It's 3 o'clock. Let's wait a couple of minutes more, and then we start. Okay. René, ¿me ayudas grabando? Ah, ya está grabando la sesión.
Sí, se está grabando.
Perfecto. Bueno, en honor al tiempo y a las personas que se han unido puntualmente a la charla, quiero darles la bienvenida a esta a este segundo webinar organizado tanto por la Asociación Ecuatoriana de Mastozoología como por la Universidad de Especialización Espíritu Santo. Les agradezco mucho por haberse conectado puntualmente. Tenemos el honor de tener como presentador el día de hoy al doctor Tomás Gillespie. How's the pronunciation, please, doctor?
Gillespie.
Gillespie, thank you very much. Con la charla La ecología de las enfermedades infecciosas, prevenir pandemias y preservar especies. Antes que nada, y como hicimos ayer, quisiera también darle la palabra y agradecer muchísimo a René Rodríguez, quien es el director de la Escuela de Ciencias Ambientales en la Universidad de Espíritu Santo, quien está colaborándonos y coordinando este evento con nosotros. Así que René, nuevamente muchísimas gracias y nos das la bienvenida también.
Muchas gracias a María Alejandra. El honor es de la universidad. Como bien dije ayer, auguro muchos éxitos y viendo la calidad de presentadores con los cuales nos han aceptado la convocatoria, creo que está garantizado y no más indicar de que a esto nos debemos como universidad, a brindar estos entornos de aprendizaje para la formación de personas y ponerlos a nivel de su tiempo en relación a la actualización de conocimientos. Sin más, bueno, démosle la palabra al presentador de esta tarde y muchos éxitos.
Muchas gracias, René. Entonces, bueno, la moderadora del evento va a ser la doctora Estela de la Torre. Ella es coordinadora del grupo de especialistas de primates de la Asociación Ecuatoriana de Mastozoología y quien ha coordinado también que se dé paso y que se llevara a cabo esta charla. Entonces, Estela, bienvenida. Muchas gracias también por aceptar la invitación y te dejo como moderadora de la charla.
Gracias, Alejandra. Buenas tardes con todos. Bienvenidos. Estoy segura de que van a ser testigos de una excelente charla. Para mí es un honor presentarles a Thomas Gillespie. Él es profesor de Ciencias Ambientales y Salud Ambiental en la Universidad de Emory y también en la Rollins School of Public Health. La investigación se enfoca en las interacciones entre cambios ambientales antropogénicos, biodiversidad y la ecología y emergencia de patógenos en especies de vida silvestre, animales domésticos y gente. Para responder a sus preguntas de investigación, Tom y su equipo usan diversos sistemas de estudio de patógenos con metapoblaciones de mamíferos, de primates, roedores y murciélagos, y vectores como los mosquitos, pulgas y garrapatas, en diferentes localidades en el África subsahariana, en Madagascar y América Latina, con diferentes grados de perturbación, por ejemplo, tala selectiva, fragmentación, agricultura y turismo, combinando epidemiología, ecología molecular, ecología del comportamiento, muestreos sociales y clínicos y modelos espaciales. Además de contribuir a entender el efecto que los cambios antropogénicos en los hábitats tienen sobre la dinámica de los patógenos, su trabajo facilita la detección temprana de nuevos patógenos que podrían ser una amenaza para la salud global y o para la conservación de la vida silvestre. Tom fue uno de los primeros científicos que demostró que el impacto humano sobre el ambiente puede alterar la dinámica de los patógenos naturales de la vida silvestre y crear oportunidades para que algunos patógenos salten de una especie a otra. Sus trabajos en Tanzania, Madagascar y Uganda son referentes importantes para estudios con la perspectiva de una sola salud y han guiado esfuerzos internacionales para proteger a especies amenazadas de enfermedades humanas y prevenir futuras pandemias. Tom es un experto externo del Foro de Alto Nivel de Políticas sobre Desarrollo Sostenible, el revisor experto del Programa de las Naciones Unidas para el Medio Ambiente, para la Plataforma Intergubernamental Científica y Política sobre Biodiversidad y Servicios Ecosistémicos. Tom, muchas gracias por tu disposición a compartir con nosotros tu trabajo. Es todo tuyo el auditorio. Les recuerdo, esta charla tiene una duración de 40 minutos. Después de los 40 minutos tendremos una sección de 15 a 20 minutos de preguntas. Si ustedes tienen preguntas, por favor, escríbanlas en el chat. Al final de la charla de Tom, Tom las va a responder. Con esto, muchas gracias, Tom.
Thank you, Stella. Thank you, Renee. Thank you, Alejandra, and many thanks to the AEM for the invitation to be with you today. Please let me know if I'm speaking too quickly or if you're having trouble with the connection. But I've put the slides in Spanish to help facilitate the discussion. So what I wanted to talk about today are two really important issues that are really of major concern today in light of the current pandemic and the sixth extinction event that we're all experiencing. Both of these involve infectious diseases, but the dynamics at play are quite different. Those that are leading to the greater endangerment or extinction of species, and the cases of new diseases emerging from nature and impacting humans. My role as a disease ecologist is at the interface of these issues. And so I wanted to talk a bit about the different approaches that we take to answer questions related to preventing pandemics and saving species. Is it sounding good? The sound quality and everything? Okay, great. So we'll start by thinking about emerging diseases. And I think most of you probably know by now that the majority of emerging infectious diseases are zoonoses. And we now know in the past decade or so that the vast majority of these emerging diseases that are zoonotic are coming from wildlife. And we also know that most of the action of emergence in the future will be occurring in the tropics. When you look at a lot of papers that have been published using available data for hotspots for disease emergence, it paints a different picture. Those heat maps will often light up in Europe, the United States, Japan, places where there's been a tremendous amount of surveillance. But it's not catching a lot of the actual emergence events that are happening where surveillance has occurred to a lesser extent. So we need to be putting more effort into looking for pathogens in the places where they are most likely to occur. So we need to start by thinking about what we do and don't know about the diversity of pathogens. And just as we know very little about the overall diversity of life at the moment, so we have the best estimate coming from Camilo Mora and his colleagues almost a decade ago at 8.7 million eukaryotic species. But with also a recognition that based on their species accumulation curves, the vast majority, almost 90% of those eukaryotic species are yet to be discovered. If we look to the case for the prokaryotes, where many of the pathogens are going to be taxonomically based, whether we're talking about bacteria or viruses, we're talking about more than 99% remaining to be discovered. And as we all know, diversity begets diversity. The tropical rainforest having the greatest overall diversity of life are also the places we expect to see the greatest diversity of these pathogenic forms. And so this is where we need to be looking if we're going to find many of these pathogens that are likely to emerge. But we need to do more than just look for pathogens. We need to understand the dynamics that are leading to their release from nature. Because it's not that nature is the problem. The problem is the way in which humans are impacting nature that's leading to a greater transmission of pathogens between species or spillover events. And so the work that we do is focused in the tropics. We do also have work in Latin America. Unfortunately, when we converted this to PDF from PowerPoint, it has blocked Latin America from the map. But I see that I have some of my collaborators on the call here from projects where we are working in Argentina, in Mexico, in Costa Rica, and other countries in Latin America. But as Stella mentioned, the core of our work has for the longest period been in sub-Saharan Africa and Madagascar. And these are places where we have been able to take an integrated approach to understanding the factors that lead to spillover events. And so this issue of disease emergence, we need to look in the places where things are likely to emerge. We need to better understand the factors that lead to spillover. We're not going to be able to characterize all of that unknown diversity of pathogens in time to stop the next pandemic. We have to find generalizable factors for categories of pathogens that have various life cycles leading to human exposure if we're going to avoid the next pandemic. And that is going to require changing some of our land use patterns. And we'll talk more about that later. The other side of this dynamic is the endangered species that are being impacted by infectious diseases. And it's only, again, in the past decade or so that people have really been focused on disease as a conservation issue. There are natural pathogens that impact endangered species. I do a lot of work with apes, with gorillas and chimpanzees. Both of them are impacted by Ebola. not coming from people. It's naturally occurring in the system. Their exposure may be greater now than it has been in the past because of human-induced factors like climate change and how that affects phenology of foods that they're feeding on and their interactions with reservoirs of the Ebola virus. But it's not something that is being transmitted from people to them. Similar scenarios with, there's a tropical form of anthrax that has impacted both gorillas and chimpanzees as well. Not coming from people, but we may be playing a role in the reason we're seeing more impacts in their populations. The other part of this is that we actually have good evidence of pathogens coming from people and impacting wildlife. And in our own work, there's been a lot of focus on respiratory pathogens and enteric pathogens that are coming from people associated with research, tourism, or other activities where they interface with wild primates, and where we're seeing well-known pathogens of humans leading to outcomes of that are impacting populations of wild primates. And so in our work, we try to address both sides of this equation in terms of spillover events. And we do this by using an integrated approach that brings into consideration socioeconomic and epidemiological survey, observational data of the people, the wildlife, and the domesticated animals that are often at the interface, a number of different types of diagnostics and characterization techniques for infectious diseases, and then spatial analyses, which allow us to bring all of this together. We also, in all of these approaches, are considering factors that would be typically examined in public health, so things like socioeconomic status, overall health, wealth, education, demography, but we also are looking and integrating factors related more to biodiversity and ecology, whether we're talking about the way in which people are using land and how that relates to the habitat of species of consideration, the climate, or the ecology of the interaction among the wildlife, the people, and the domesticated animals, or the way in which the wildlife is responding to the way in which we have altered the landscape, or the way in which we have altered our interaction with the wildlife. And collectively, this is a One Health approach. This is a construct that is gaining a lot of traction right now in not only how we approach research, but how we respond to these challenges. So you'll be hearing more about One Health in the future. We also need to use tools to try to think creatively about transmission dynamics, because if we go into a system and we don't know it very well, we need some way to measure the risk of transmission, knowing that pathogens are rarely transmitted. It's not something we're going to see without tremendous sample size. And so the way we can do this in a system that we're just starting to learn about is to use a model for transmission. And we often use E. coli, Escherichia coli, the common commensal bacterium, because we all have it. And when I say we, I mean all of us humans and the other mammals. So when we're thinking about transmission between wildlife, domesticated animals, and people, they all share E. coli. And so we can compare the diversity of that E. coli and the similarity of the E. coli between species, between individuals, to gauge how often we're seeing transmission between these various groups of individuals. Whether we're talking about different species of wildlife, like these 3 species of monkeys that we've examined these questions related to in Uganda, the domesticated animals, or the people in that system. And this is an approach that works well in most systems. By using this, we can see, for example, from that scenario in Uganda, that one of these species of monkeys, the red-tailed guenon, has bacteria that is much more similar to that of the people and the livestock that they overlap with compared to the black and white colobus and the red colobus, the other two monkey species. And so something is creating a phenomena where there's more of an opportunity for bacterial transmission, either through the environment or directly between red-tailed guenons and people, or vice versa. This analysis, what you're seeing here is an FST score, a measure of genetic similarity, does not give us a directionality. It just tells us we're seeing similarity between them, but not which direction the bacteria is moving. We can get at that with some other aspects, which we'll talk about in a moment. So that's one part of the equation. Then with our spatial analyses, we can start to see, we can plot a lot of our observational data to see where there is overlap and how this relates to boundaries between protected areas and landscapes that are used by crop-raiding primates, used by people, and used by domesticated animals in various ways. And we can overlay data for survey data on other aspects of where we're seeing interaction and contact. Here you're seeing an interview measuring using a tally board-based approach, where we leave respondents with envelopes with cards for various animals that may be crop raiding in their fields, and the envelopes each have the image and name of a crop that they're growing. And since they're intercropping, they probably would have 2, 3, 4 envelopes for the different crops. If they have a crop raiding event, they put the card of the animal that they believe did the crop raiding in the envelope corresponding to the crop that was impacted. And they put the day of the month on the back of the card. And so by doing this, we get really fine-scale data on the impacts that are happening in ways that we often would not get from a survey alone. If we go and do a survey every month, there's not good recall of that information. Just as you may not remember what you had for lunch last Tuesday, or what you wore to work or to school last Tuesday, it's the same in thinking about these crop raiding events. If you interview people at the end of the month, they often only remember what were more extreme events, but the cumulative impact of smaller events is often more important in terms of the opportunities for disease transmission, or even the impact on the crop in some cases. We can assemble that data, and here we're seeing many different crops being grown simultaneously within communities. And when we look at maize here, the maize is actually the most commonly impacted crop. And the organism that is responsible for much of the damage, the most damage, is the red-tailed guenon, that monkey that was associated with much more similar bacteria to the people and the domesticated animals. When we further our survey technique to try to understand this, we learned that people are using a technique called maize daubing. They're taking the fecal material, the dung of the cattle, and they're putting it on the ear of maize to protect it from consumption by the monkeys. Sometimes they add sand, sometimes they add cayenne pepper, but this is a technique that is effective at keeping the monkeys from feeding on maize. Unfortunately, they still investigate it. It provides an opportunity for the monkeys to become infected with human bacteria, or more likely with the bacteria of the domesticated animal, because it's direct contamination of the feces of the cattle. It also helps us to understand why the bacteria of the people is so similar to that of the domesticated animals, because it's very easy for them to infect themselves accidentally while they're preparing the maize to avoid crop raiding. We've used the same approach with E. coli as a model to look at how tourism and research can lead to disease transmission. And in this case, we've done this with chimpanzees and gorillas. And as we saw in the case of the monkeys, the types of interaction that lead to proximity in a shared environment are the most telling in the similarity of the bacteria. In the case of the mountain gorillas, here I'm showing you this example. There are gorillas that have a lot of human exposure. The ecotourism gorillas happen to also spend half of their time outside of the national park. They interact with tourists. They also interact with researchers. So they have a lot of interaction with many that could contaminate them with environmentally mediated bacteria. Then we have research gorillas who are typically only exposed to researchers, and what we call wild gorillas, which are gorillas that live in the core of the national park and rarely have overlap with humans. And when we look at the similarity of the bacteria of these various gorillas to the gorilla guides, the research field assistants, people living in the villages around the national park, and the domesticated animals, in all cases, we see that the bacteria of those ecotourism gorillas is most similar to each of those groups. And as we look at gorillas that have less interaction, less overlap with people or domesticated animals, their bacteria is far less similar. So again, suggesting that this overlap, this interface is leading to frequent transmission of E. coli between these species. And then to try to get at the directionality of this, we can look at something like the prevalence of antibiotic resistance. And what we see is that there is a high proportion of resistance in gorillas to commonly used antibiotics in the system, but there's no resistance to antibiotics that are not commonly used in the system. It's important to know that gorillas are not being given antibiotics in the system. And so when we find antibiotic resistant bacteria in gorillas, it suggests that they have been exposed from people or domesticated animals. And from our work in local pharmacies and from our survey-based approach, using pallets of drugs as well as asking for the packaging of completed drugs and the names of drugs used, we can see which drugs are commonly used and which aren't. The resistance levels that we're seeing in the mountain gorillas that are most exposed, those ecotourism gorillas, is the highest level that we have seen in any wildlife that we have examined. So that gives you a sense of how we can go about seeing how transmission is happening in a system where we don't have a good sense yet of how it's happening. Once we start to understand where the transmission events are occurring, we can start to look at known pathogens. So going beyond the model and targeting pathogens. So that's what we've done in Tanzania, in Gombe National Park. So we've built upon the long-term work of Jane Goodall, celebrating 60 years of behavioral research with the chimpanzees this year. And for the past 14 years, we've been looking at disease dynamics in the chimpanzees at Gombe, and we've integrated this One Health approach to also understand how this interface relates to what's happening in local people and domesticated animals, as well as other species of wildlife, particularly baboons. And so we use, again, this mixed methods approach where we're sampling all of these various organisms that are interacting we're sampling the local human community, we're collecting the spatial data, the survey data, the observational data, and we're also sampling some of the environmental mechanisms for transmission, like the water, soil, that could lead to indirect transmission. And we've focused in this system on a number of pathogenic enteric bacteria, things like Salmonella and Shigella and enterotoxigenic E. coli, some of the toxic strains of E. coli. And by looking at these organisms in the system, we can see high prevalence of some of these across the board, like the enterotoxigenic E. coli. We can see that Salmonella is occurring in the domesticated animal, the wild primates and people, and Shigella is occurring in people and the wild primates. So there's a lot of potential for cross-species transmission of these important enteric pathogens. These are things that can cause severe diarrheal disease or even dysentery. When we characterize them more directly, here we're seeing Salmonella, we can see that the linkage is far more important between people and the wild primates than with the domesticated animals. We don't see the same dynamic of domesticated animal-human similarity in bacterial transmission in Tanzania at Gombe that we saw in Uganda. When we look at Shigella, this is both Salmonella and Shigella are important sources of bacterial dysentery. Here we see really strong support for cross-species transmission between humans and wild primates, both chimpanzees and baboons. And when we look at antimicrobial resistance in this system, we are able to test against the specific pathogens. So looking at resistance to Salmonella, resistance to Shigella in the system. And we're seeing very different, which suggests that the selective pressure for resistance is distinct between these organisms, meaning that people are using antibiotics in different ways that can impact how we would deal with this for the conservation of the apes and for helping the local human population in the future. So we're using very similar approaches to understand dynamics in Madagascar, where we're looking at the interface for local human health and lemurs. And in this case, domesticated animals play an important role, as well as peri-domestic animals, things like rats and mice, which are moving in and out of homes and interacting between people and wildlife. In the system in Madagascar, I just want to highlight, we don't have time to get into a lot of the details, but just to highlight the different skill sets that have to come together to answer these types of questions. So you're seeing veterinarians, you're seeing ecologists, you're seeing anthropologists, geographers, nurses, doctors, students of public health, students of development. All of these are critical fields to be able to answer these types of questions. We need to bring people together to work together on these questions. We also need better linkages between empirical scientists and theoretical scientists. So you'll see many papers published in journals like Science and Nature that are based on taking data from previously published work and then mining that data to create elaborate models. But those models are only as good as the data that goes into them. And so we're often dealing with data deficiency, or a case where those who are creating the models don't fully understand the variables at play or the quality of data related to those variables. So it's really important to get people to work together. And we've been able to do that with help through NCs and CSync to create groups of empirical and theoretical scientists working together on how land use change can affect infectious disease dynamics, for example. By doing this, we're able to see some important generalizable patterns, like when you fragment a system, once you hit about 40% disturbance of the system, that's where you have the maximum opportunity for spillover to occur, for disease emergence. And then once you hit something like 80%, the vast majority of that risk is gone. And so during that process of transition, we can target where we're likely to see emergence occurring. And that helps us actually put a cost on disturbing systems. Because right now, banks are loaning money. The World Bank is going forward with projects that can have a tremendous impact on natural systems and that could lead to spillover without accounting for that cost. So through this work, we've actually been able to guide the process. It's led to a paper many of you may have seen in the past few weeks in Science that's actually putting a price on pandemics and really focusing on the role of large-scale domesticated animal industrial farming and deforestation as critical factors in leading to disease emergence. And being able to put an economic price on this and showing that that is very small, it's 2 orders of magnitude smaller than the cost of the current pandemic that we're dealing with, for example, is helping to lead to global change in how we think about financing large-scale land use projects, how we measure the potential impact of these things, so we can make better choices for a more sustainable future that has space in it for the biodiversity that we all care about. We're also doing a lot of work in terms of helping to reduce the risk of exposure in wildlife to these common human pathogens. And so 5 years ago, we released best practice guidelines for protecting the great apes from infectious disease and monitoring great apes for infectious diseases. And so these guidelines really focus on preventing infections from getting into these wild populations, have surveillance that's able to look for these things, and also how do we go about clinical interventions? When should we do an intervention? When shouldn't we? The kinds of things that are advocated here, some of the simple things that are quite effective are the same things we're talking about with the current pandemic in humans. We need to wear a mask, we need to keep a distance, and we need to be vaccinated. So there are a number of human pathogens we know can have a tremendous impact on wild primates. We need to vaccinate against them. And then we need to think a bit further about employee health programs and community health programs. We need to have collaborations with ministries of health to work on community health at the same time that we're working on protecting wildlife from disease. With the COVID-19 pandemic, we know that our guidelines don't go far enough. And so we have developed further guidelines in collaboration with the Wildlife Health Specialist Group of the IUCN. And we've also released a paper in Nature really focused on why this is so important and really trying to focus international attention on the issue. Many of you probably know that many of the primates are considered to be very susceptible to the SARS-CoV-2 virus that's responsible for COVID-19, and that much of this data is based on the similarities in the initial binding site, ACE2 binding site of humans and other primates. And you probably also heard that the neotropical primates have less similarities in their binding site and therefore are considered less susceptible compared to the apes and the African and Asian monkeys. And then lemurs are considered to be a mixed bag based on the data that's currently available. But it's really important to recognize that that initial binding site is just one part of determining susceptibility or how impactful the virus could be in these populations. So if we look at mink, for example, they have a very low binding site affinity for COVID-19. So we wouldn't, based on that alone, we wouldn't expect mink to commonly become infected. But based on the data, we're seeing that mink are actually becoming infected. They're culling large numbers of mink in Europe. And we've seen mink to human transmission as well in these industrial farming facilities. And so this just highlights that we need to be cautious in assuming that this initial binding site alone is the best indicator. We should be careful with all wildlife until we have a better understanding of the overall risks. And some of these other factors that can be also critical in determining susceptibility and how impactful a pathogen can be in a given species or population of that species. We have created a hub for information for COVID-19 and non-human primates. And so I encourage all of you to go here. There's new information every day on what we know and what we don't know. This is primarily focused on primates, both wild and captive, but there's a lot of information and data as well for other species of wildlife, as well as people. So I encourage you to go to this site, learn more about the data that's available, and also efforts to try to mitigate the impact of COVID-19 on us and other species. And then lastly, I want to highlight that there are also guidelines being put forward more broadly for wildlife beyond primates. And this is a collaboration of the Wildlife Health Specialist Group and the OIE within the United Nations to have just broad categorical recommendations for mitigation of COVID-19, and doing that by minimizing exposure risk, being able to assess this, and to protect. So I want to stop there in terms of time so that we have plenty of time for questions. And we can talk about whatever is most interesting to those that are joining us today.
Muchas gracias. Súper interesante tu charla, todos los temas que has tratado. Vamos a esperar un par de minutos para que la gente que tiene preguntas, por favor, las escriba en el chat. Tenemos ya un par de preguntas. La primera, Carlos Carrión, asks you, what can be done to avoid the next pandemic? How you think One Health will take this question?
So there are two really important parts to that, to answering that question. Thank you, Carlos. The first is we do need better surveillance, not just of wildlife, but of wildlife and people and domesticated animals at this interaction point. So in the last 10 years, there have been tremendous efforts by groups like PREDICT through USAID and others to catalog more of that diversity of pathogens that are out there. Despite all of that effort, they've only categorized less than 10% of the viral pathogens of mammals alone. And we all know, us as a group focused on mammals, we know that there are not that many mammals. There are just over 5,000 species of mammals total. And so they're a very small slice of the diversity of life. Yet, after 10 years of effort trying to focus on their diversity, pathogens, less than 10% have been accounted for. So we need a better job doing that and integrating surveillance of people and domesticated animals at the same time. The second part is that we know that disturbing systems in the most diverse places, like tropical rainforests, will lead to a greater risk of emergence than many other activities. So we have to reduce deforestation in these areas. We have to reduce things that are leading to unique types of contact between tropical species and people. And so part of this, one thing that's happening right now is a greater emphasis on how financial decisions are made for actually funding the upfront cost of large-scale land use change. So if you're talking about oil palm plantations, soy plantations, types of large-scale agricultural efforts that are leading to dramatic land use change, there needs to be more accountability for spillover as a potential cost to those activities. And then we need to be making personal decisions that for some of those goods. We've heard recently in the news about the link to livestock production as well as an additional factor.
Muchas gracias, Tom. Martin Kovalevsky says, hi, Tom. What is your advice about continuing fieldwork with neotropical primates during COVID-19?
So the ethical conundrum of how do we move forward with fieldwork is central to all of us. I mean, no one wants to lead to exposure events. The good news is, this many months into the pandemic, we haven't yet had documentation of primate exposure in the wild. People have purposefully infected primates in captivity, and that has demonstrated that in macaques, they become ill like people, and so there is a susceptibility. The only neotropical primate that I know of that they have infected purposefully is a species of marmoset, and it was not viable. And so, at least in that case, there appears to be a lower susceptibility. The risks are also highest in terms of the proximity. So we know now, early on, we were much more focused on exposure through shared environments, right? The idea that the virus could be shed into the environment and then you could have later exposure. It seems that proximity is far more important based on the data from people and the exposures that have occurred to other species in captivity. So because of that, terrestrial primates, terrestrial wildlife in general would be more at risk than arboreal because of the proximity and the shared environment. But what we're recommending is there are guidelines that we put forward for those who are doing research with apes, as well as those that are involved in surveillance for law enforcement in those areas, or those that are involved in extractive industries that overlap with the habitats of wild apes. And most of those guidelines are very much focused on wearing a mask to varying degrees depending on proximity or whether or not you're in the habitat and the role of the individual. Minimizing the number of individuals that are moving in and out of the habitats, and avoiding close proximity to any wildlife. Those are the overall recommendations. But a lot of us are just being very cautious. So we're holding back. We're trying to make sure that there is enough of a presence to ensure that other things that researcher presence are leading to a reduction in, like poaching or other impactors on the species, are not occurring more because people are too far back. So it's difficult. It's a complex decision to make. But I would suggest going to the IUCN Great Ape guidelines as a starting point.
Gracias, Tom. Richard Bobo says, can you tell us about neurological diseases in primates and the possible contagion to us humans?
Neurological?
Yeah, neurological. neurons, nervous system?
Urinary.
No, nervous.
Nervous.
The brain, the nerves.
So maybe thinking of prions, or Richard, do you want to clarify more specifically? Is there an example of a pathogen that you're concerned about?
He says brain diseases in general.
So yeah, I don't know a lot about examples like that, but I see Julio is saying maybe West Nile would be something to think about in this regard. Yeah, I haven't seen a lot of research on that topic.
Thank you, Tom. Alexandra says it strikes me that there is this transmission of diseases between apes, such as gorillas and humans. I would have imagined that one cannot be that close to these animals, especially tourists, but I can see that they can. How this interaction between tourists and apes occurs? Can you touch them? Can you feed them? I wonder what the tourism business is like. Are there any special regulations?
That's a really important question, Alexandra. So tourism is a double-edged sword in many ways, right? So it's necessary. It's actually provided for the conservation of many species, especially the apes. They're flagship species in that way. But at the same time, we know that there has been exposure to respiratory pathogens, for example, that have led to major mortality events through that same dynamic. There are regulations. The best practice guidelines are the starting point for that process, and then each country is deciding for itself what the regulations are. And so the best practice guidelines are followed to varying extents depending on the country. And then there are also issues of whether or not the rules at the local level. And often one of the big challenges is the economic imbalance between the tour guide and those that are tourists. So we often see wealthy people who are visiting, and they're offering large tips to try to get permission to break the rule, to go closer, to be in the area when they are clearly symptomatic of infection and so on. So it's a constant effort to try to reduce the impact and to also help with some of these issues of economic imbalance.
Gracias, Tom. And Daniel says E. coli is a great model for studying the direction of the infection because of its resistance to antibiotics. But in the case of viruses, what difficulties are there to track the direction of the infection when you use genetic sequences?
That's a really important question. But in general, many of the viruses are the most difficult to be able to examine transmission pathways with, because they are often acute infections, and so you're very unlikely to catch an active infection in space and time. And people often resort to antibodies to provide a reference of previous exposure. The challenge is the specificity of some of those antibody tests are not well validated for wildlife. And so it's hard to interpret what antibody results really mean relative to when infection occurred or if infection ever actually did occur. And so instead of focusing on virus versus bacteria versus parasite, we try to focus on the life cycle, the mechanism of transmission. So thinking of it more as respiratory or waterborne or direct transmission or environmentally mediated contamination, because we have good examples of these that are chronic, bacterial, or parasitic, that can also give us insights into what we would expect with an acute viral pathogen.
Thank you, Tom. We have a couple of comments. Julio Cesar says, great talk, great research and talk, Tom, congratulations. We have also thanks from Carlos Carrion. I don't see any other question. So I guess that would be it. Thank you very much, Tom. Alexandra, I don't know if you want to host this session, this talk.
Yes. Thank you very much, Tom. Thank you for answering my question as well. This video, the video of the talk is going to be available for everyone who couldn't be with us today in the AEM web pages and social media. So be aware of our announcements. So thank you very much for sharing your time with us, Tom. That was an excellent talk, very interesting. Daniel Chavez says, very nice talk. Thank you, Tom. And thank you, Stella, as well, for helping us for be part of this for this event. So yes, that's all. So solamente quiero decir al resto de asistentes que el día de mañana continuamos con las charlas para que nos acompañen también. El día de mañana estarán con nosotros el grupo de especialistas en murciélagos del Ecuador y la charla será sobre el proceso de producción, edición e impacto de la enciclopedia Handbook of the Mammals of the World y estará a cargo de los doctores Sergio Solari y Ricardo Moratelli, que son especialistas, taxónomos sistemáticos, muy expertos en murciélagos, sobre todo neotropicales. Again, thank you very much. Thank you all for being here with us. Estela, gracias también. Y nos vemos en una próxima ocasión con todos.
Gracias, Alexandra. Solamente si puedo terminar con una cosita que yo creo que es importante que, y espero realmente, y esa fue una de las razones por las cuales en el GEPE quisimos hacer una charla con Tom, es llamar la atención de que en nuestro país no tenemos prácticamente nada de información sobre estos temas. Vivimos en probablemente el lugar más biodiverso del planeta, en una zona tropical, en una zona, de acuerdo a lo que nos acaba de decir Tom, donde hay una altísima probabilidad de que tengamos la emergencia de nuevos patógenos, de enfermedades zoonóticas. Ojalá la gente que ha estado en la charla también se motive para empezar a estudiar esto. Muchísimas gracias, Alexandra. Felicitaciones a la AEM y muchísimas gracias. Estamos en
All right. Thank you, everyone. It's been a great opportunity to talk with everyone today and to hear about some of the questions and issues that you're thinking about. There are many people who are really interested in dealing with some of these dynamics in Latin America. I can see, some of these good friends and collaborators are on the call today. So I'm happy to see this discussion continuing, and looking forward to where this can go from here. Hopefully, I can be in Ecuador soon to be able to talk to one more in person.
Yes, hopefully, yes. So be safe, take care, you all, and I hope to see you very soon.
Muchas gracias.
Gracias.
Thank you very much.
Thank you very much.
Excellent talk.
All right.
Para salir solo aplasto, me salgo y me voy, ¿sí, no?
Hay una, como otro menú en la parte izquierda,
Ya.
superior izquierda. Ahí
Bien.
dice cerrar sesión, ahí se sale.
Gracias, nos vemos.